Welcome to the Longevity Mindset channel, the channel that helps you discover why you should want to live a long, healthy, wise, and happy life. Here we go. Let's get started right away. Look at these two little mice for a second. They're twins, but incredibly one aged much earlier than the other. How is this possible? Simple. Scientists managed to turn back the hands of time. In this video, I'll explain how it works and update you on the many recent discoveries related to aging. Welcome once again to the Longevity Mindset channel, a place dedicated to sharing ideas and strategies for living a long, healthy, wise, and happy life. But first of all, don't forget to subscribe to the channel right now to stay up to date with all upcoming releases. Okay, now let's get back to the topic. The news we're discussing concerns two recently published studies in Cell and Nature, two prestigious scientific journals. Everything I'm about to share with you is the result of decades-long research. Behind these recent publications is a group of researchers led by David Sinclair, a professor at Harvard Medical School. He is also the author of a highly successful popular science book titled Lifespan. The astonishing and confirmed fact is that researchers managed to reverse the aging process in laboratory mice. The biological age of the mice regresses by several months, which in humans would correspond to several decades. No special effect, no black magic, just a lot of scientific research. By watching this video, you'll understand the science behind this astonishing result and the scenarios it opens up for humanity. The starting point for aging research comes from the studies of Professor Hayflick of Stanford University in the early 1960s. He invented the first microscope, which allowed direct observation of culture plates and live cells. Thanks to this invention, Hayflick could observe how cells from various organisms multiplied. He made a simple but fundamental discovery. Cells from an organism, when cultured in the laboratory, age. Cells can only undergo a fixed number of cell divisions, after which they stop dividing, enter senescence, and the cell culture no longer progresses. The same happens inside our bodies. They age because over time our cells lose their regenerative capacity. This limit is also known as the Hayflick limit. Human fetal cells can divide 50 or 60 times in culture before stopping. From Hayflick's early studies to the present day, there have been numerous research and theories about aging. In summary, it's unlikely that there is a single phenomenon that explains aging. But it's also plausible that the different known causes are not independent of each other, and some mechanisms are hierarchically superior and therefore more influential. Let's focus on some recent discoveries and things we all know but you might not have connected to aging. First clue, think back to when you were little. Now go back a bit further, a bit more. Think about when you were fertilized eggs. Your parents conceived you. Maybe they were already adults of 20, 30, or 40 years at that time. Both your parents' cells had already undergone many divisions. But what's extraordinary is that you weren't born with the biological age of your parents. Your biological age started from 0. You were made entirely of new cells with intact reproduction potential. This means that something happened during fertilization that reset the clock. During fertilization, the genetic program of the cells starts from the beginning, starts from 0. It's something we take for granted, but it has significant implications in aging research. It has always puzzled regeneration researchers because there's evidently a yet-to-be-discovered key to understand what happens during fertilization. Whatever the process of cellular aging might be, we know that going back is possible because it happens every time a new individual is born. Another clue comes in the form of Dolly the sheep in 1996. Dolly, the first cloned mammal, was born followed by many others. To clone an animal, you take the nucleus of an adult cell, usually from the skin, and insert it into an egg cell that has had its nucleus removed. The embryo that develops has only one DNA, that of the donor, because the nucleus comes from the donor. It's interesting that when we clone an animal, the resulting clone has the same DNA as the donor, but doesn't age at the donor's rate. If we clone an older animal, we still get an embryo, and eventually a cub with cells that start with the entire division potential. Clones don't age prematurely. Even during the cloning process, the age clock resets, just like during fertilization. How does this mechanism work, and how do we go back in age? Where this odometer is located is not entirely clear yet. But today we know, thanks to studies on clones, that the biological age counter isn't in the DNA sequence, which remains the same between the clone and the donor. The biological counter is in the epigenetic information or epigenome. What's the epigenome? It's all the genetic information not directly written in the DNA letter sequence, but depends on reversible modifications of chromosome structure. A typical epigenetic modification is called methylation, in which small chemical groups called methyl groups attach to DNA like labels that can be glued, removed, or reattached. Metaphorically, when these chemical groups attach to DNA, it prevents its reading, like a book with glued pages where the instructions written inside the pages cannot be read. The instructions in DNA are always there, but if that information isn't readable, it's as if it isn't there, and the cell doesn't know what to do, leading to disorder and aging. This creates a situation of entropy or disorder that gradually leads to cell deterioration and loss of regenerative capacity. This simplification represents the dynamics of the phenomena that collectively determine what we commonly call aging. The good news is that the information isn't lost, just like the instructions on the book. This also applies to the information contained in DNA, which is still there in its place. It's as if every cell in our body contains a backup copy of what we were as embryos or children. If we could reopen the pages of the book, the information will become readable again. And in fact, this is what happens, as we saw earlier, during fertilization and cloning. The DNA is accessible to the cell again as if it were new. If we know how to reprogram the epigenome and understand what deteriorates over time, the biological age clocks can be turned back. Yes, it sounds like science fiction, but it's what we are already capable of doing today. The mastermind was Shinya Yamanaka, a Japanese researcher and the author of an extraordinary discovery. Yamanaka was looking for a way to reset adult cells, the ones in our bodies, for example, in the skin, to revert them to a stem cell state, just like the ones found in embryos. He wanted to reproduce what happens during fertilization and cloning, but without fertilizing or cloning anything, simply by encouraging cells to reset on their own. After trying countless combinations, Yamanaka discovered that this could be done surprisingly simply. All it took was to provide cells with a cocktail of 4 proteins, or the genes that produce them, called MYC-OCT3/4-SOX and KLF4, now commonly known as Yamanaka factors. Thanks to this incredible discovery, we can now take adult cells, such as skin cells, treat them with the 4 Yamanaka factors, and reset their epigenetic program, convincing them to become stem cells again, capable of transforming into other tissues, such as muscle or neurons. Yamanaka's system is the magic that turns back the epigenomic clock. In 2012, Yamanaka rightfully won the Nobel Prize together with the Englishman John Gordon, and his system is now used in labs worldwide to obtain induced stem cells from adult cells. Yamanaka's system is fantastic when applied to cells, but when it comes to rejuvenating an entire human being, there's a problem. It works too well. The reset is almost total. The system turns cells back to the beginning. It transforms them into baby cells and erases their identity. A skin cell treated with Yamanaka's cocktail is no longer a skin cell. It resets its genetic program, ready to become something else. It's no longer a differentiated adult cell, but becomes a pluripotent cell like the ones found in embryos. Many are fascinated by the idea of rejuvenation, but no one wants to become an embryo again and start from zero. Our cells need to rejuvenate, but without losing their identity. Otherwise, the whole body falls apart. Muscle cells must remain muscles, skin cells must stay in their place, and so on. We want to preserve our identities, our memories, our affections, everything we are and have learned. The idea isn't to be reborn or create a younger clone of ourselves, which would still be another person, but to merely turn back the biological clock just enough to stop or delay the degeneration process associated with old age. It seems impossible to have it all, but apparently it is not. Now it's time to return to David Sinclair and his colleagues and their recently published studies, because we finally have the basis to understand their discovery. To reverse aging in their mice, Sinclair and his colleagues used the Yamanaka cocktail, but shortened it. One of the 4 factors is missing, specifically the MYC. They discovered that without MYC, the Yamanaka cocktail, OCT3/4-SOX and KLF4, or OSK as it is now called, works the same way but without erasing the cell's identity as the complete cocktail does. With the new shortened cocktail, the epigenetic clock resets and the cells rejuvenate, but they maintain their identity. But what are the tangible results of these discoveries so far? The experiments focused on the effect of the OSK cocktail on the optic nerve. This is because we know that the optic nerve of young laboratory rodents regenerates if damaged. But this ability is lost in adults. This is why the optic nerve is a widely used model for studying neuron regeneration. In adults not treated, where the cocktail is not activated, the optic nerve does not regenerate if damaged. However, in those where the OSK cocktail is activated, the optic nerve regenerates, and the animals regain their sight. In conclusion, where will these fantastic scientific discoveries lead us? These are certainly preliminary results, but they are extremely interesting, and hint at developments with consequences that are hard to fully comprehend. These initial results confirm that epigenetics is one of the most important and high-level avenues for understanding aging. A system that reverses the aging process in human beings and can be repeated cyclically, say every 20 years. In theory, it could go on indefinitely for who knows how long. But let's stay grounded. The realistic idea is not to become immortal or to live for 300 years, but to delay the process of organism degeneration we call aging. The goal is to spend more years, perhaps our entire lives, in full physical and mental capacities. Healthy and active, accumulating experience and wisdom. Evolution is exponential. In recent decades, we've sent spacecraft to Mars, decoded the human genome, and managed to coexist with artificial intelligence. What else could happen in the next 20 or 30 years? Professor Sinclair himself stated that scientific developments are much faster than he could have expected, and many of today's discoveries could become reality much sooner than anticipated. It's not about fighting old age at all costs or wanting to eliminate the elderly, but about living the time we have to the fullest, developing consciousness, and contributing to the evolution of the whole. Are you ready to rejuvenate and have much more time at your disposal? Now is the time to start imagining how you would like to invest the extra time you will have at your fingertips. Thank you for watching the video. If you enjoyed the content, please leave a like, share the video with your best friends, and subscribe to the Longevity Mindset channel so you don't miss upcoming updates. We look forward to see you in the next video. See you soon.